which I've done now, so we're recording. Welcome everyone. The Queensland Mycological Society acknowledges the traditional custodians of country throughout Australia and recognises the continuing connection to lands, waters, fungi and communities. We pay our respect to Aboriginal and Torres Strait Islander cultures and to elders past, present and emerging. As I'm speaking to you from the Sunshine Coast, I would also like to acknowledge the Gubbi Gubbi people in particular, who are traditional custodians of this land. So welcome everyone. As you know, the Queensland Mycological Society is a group of citizen scientists. And what I usually observe about citizen scientists is they have this common value of being curious and engaged. And we love that. We love that you are observers and of fungi. And in that sense, we really love those people to take the leap into citizen science. And of course, the first leap into citizen science is a very easy step to take, and it's really about becoming a reporter. So when you're a reporter, basically that means whatever you find out in the field, get online on social media such as Facebook, get on one of our pages, QMS or SEQ Fungi, and share what you find. And it's a really simple thing to do. Just get online, show and tell. Say, hey, I found this, and that means we can act on something that's interesting or get a feel for what's out there at the moment. So we really do need a lot of eyes on the field, gathering data and reporting their findings about what's coming up where. So be a reporter. I imagine most of you are on Facebook, and that if you're not, I encourage you to get on a social media platform and share your finds. The next logical step would be to be a data collector. And a data collector is someone who actually goes out in the field and records data. It's as simple as downloading the iNaturalist app and uploading your photos to the iNaturalist app and recording the location and image data that you find. The purpose of this talk today is really for the people who are in the next stage of citizen science, so people who are specimen collectors. So our intention in this talk is to provide you with valuable information, in particular about polypores that you find in the field, and that if you are collecting those specimens, or even if you're just collecting data on the app, what sort of images you need to take and what sort of information you might need to collect about polypores. And of course, the next step for a specimen collector would be to work or volunteer at a herbarium, which means they would then be able to work on specimens and help lodge them at an appropriate repository such as the herbarium. And the end game, of course, is to specialize in a group of fungi. And that's what Matt Barrett does. Matt's been specializing in polypores for over 20 years now. So he really does have a good feel and an excellent take on polypores. And he really is the go-to with regards to getting assistance with working on polypores. So he has a lot of value to add in this space, and in particular to these people, the specimen collectors and the data collectors. So I'm going to hand over to Matt now. So Matt, if you wanted to share your screen.
Thanks, Wayne.
Welcome.
Thank you. All right, can you all hear me?
Yes, we can hear you lovely.
That's great. Good day, everyone. Thank you for inviting me along today. I'd like to start off by acknowledging the Yuganji Indigenous traditional owners of the Cairns area where I'm speaking from. I'm Matt Barrett, for those of you who don't know me. I work at James Cook University in Cairns, at the Cairns campus, at the Australian Tropical Herbarium. And I'm going to be talking to you today about polypore diversity in Australia, and hopefully giving you at least some pointers towards identifying some of your bracket fungi. But I'll also be giving you some idea of just how difficult it actually is. Okay, so the aims of my talk today are to give you a little bit of an overview of my research, but to give you a bit of an idea about the diversity of Australian polypores and give you some familiarity with some of the key traits that you need to know to identify them. And hopefully this will improve your photography of those critical traits, particularly if you're uploading things for iNaturalist or sending photographs for things of things to be identified. And hopefully this increased experience will increase your ability to recognize differences between species and recognize more species in your area. The ultimate aim of what I'm trying to get you to do, which is upload photographs to iNaturalist and ideally collect specimens from the herbarium, is to help map and document the Australian polypores, which are nowhere near as well known as they should be and really need to be. We'd really like to improve the knowledge of host associations. So if you're able to identify the plants as well, that would be fantastic, because that connection between the fungus and the plant is critical. And ultimately, if we can identify all of our polypores much more easily, then we'll actually enable ecological and biodiversity studies. Polypores are used elsewhere in the world as indicators of forest health and indicators of biodiversity. But in Australia, we aren't able to do that because we simply can't identify most of our polypores at this point in time. So what are polypores? Well, they're more or less what you know as bracket fungi, but they have a huge array of different shapes. To make an exact definition is very difficult. They are basically things that have their spores borne on a pore layer on the underside. So if you turn the thing over, it has pores on the underside. But it also includes some of their relatives that have teeth or gills, excluding the boletes, which are very fleshy and are usually treated as a group on their own. They're usually on wood, but sometimes they can be on soil or leaves or grass, or even sometimes on other polypores. They're often leathery or woody, but they can be very soft or fleshy or cheesy or even gelatinous. Bracket fungi or shelf fungi are their common names, but many of them are not brackets or shelves, such as the one down here in the bottom right-hand corner. I hope you can all see my mouse moving around there. And then they're sometimes called crust or skin fungi, but they differ from often a lot of the other crusts and skin fungi that have smooth surfaces, because they still have those pores underneath. So I said that some of them can have gills. This is an example of Lentinus concinus, and it has gills like a normal mushroom, but it's quite tough and leathery like a normal bracket fungus. And you can see deep down inside the gills, these little cross veins, which are the remnants of their ancestors that had pores. So this is quite characteristic of some of the species of Lentinus. Over on the right here is another polypore relative that has evolved to have gills. This is a species of Panus. This is a very widespread species in Northern Australia, but as far as I can tell, it's undescribed. It doesn't appear to match any of the species from elsewhere in the world. And it has two very strange forms. One of them just looks like a normal agaric and grows on wood. The other form has lost the ability to form normal gills, and the gills actually become what we call secotioid, and the gills are all sort of munched up like a truffle, and internally they just have these locules and release their spores out into the locules. So it's a very strange thing, and as far as we can tell, there's no real difference between these two forms, other than the fact that one of them has gills and one of them has these more strange munted arrangement. So this is sort of a polypore that has become gilled. This is making a transition back to something else again. There are polypores occur right the way across the basidiomycete group of fungi. So they all bear their spores on basidia, which are cells that produce the spores on the outside of the surface, rather than the ascomycetes that bear the spores internally in the cells. There are two main orders or groups of polypores. One is the Polyporales, which usually, but not always, has white to yellow to red to orange flesh. And then there's the Hymenocutales, in particular the family Hymenocutaceae, which has this dark rusty brown flesh that can be anywhere between sort of fibrous and woody. And when you put drain cleaner on them, or potassium hydroxide or sodium hydroxide, they instantly turn black. And those are two quite distantly related groups of polypores, and you will have seen examples of both of these just walking around. There are also a bunch of polypore genera scattered across a whole range of other different orders, For example, this Amylosporus, which is a relative of Russula that has pores on the underside, and its spores are very small, but they turn blue in iodine and they have little tiny ornaments on them. And so the spores are similar in some regards to Russula and sort of betray that relationship in the family, in the order Russulales with Russula. So what have polypores ever done for me? Well, they recycle wood. They are one of the main recyclers of wood globally, and they are absolutely critical to the global carbon cycle. So as the trees are stripping carbon dioxide out of the air, eventually that tree will die, and that carbon needs to go somewhere. Otherwise it will just build up in the forest. And polypores are part of the group of fungi that break them down. They do that by having a huge range of enzymes that target the cellulose and hemicellulose and lignin. And some of them are very voracious decayers of wood. And depending on exactly how they break down the wood and how much they break it down, sometimes that wood is completely consumed and recycled for other plants to use. Other times it's broken down and part of it is left as carbon and sequestered in the soil. And brown rot fungi in particular can sequester quite a bit of carbon in the soil. So both polypores are important, and their diversity of what they do to the wood is also important. These enzymes have biotechnology uses, and there's a lot of interest at the moment in developing these enzymes for biopulping paper, bioleaching paper, and degrading plastics. So avoiding uses of some really nasty chemicals in some of the major products that we use, particularly associated with paper. Ecologically, they are very important for a lot of animals. They are primary recyclers of wood, as I said. They are assisted by termites and beetles, and they often coexist in the same space. This is an example of a beetle that has co-evolved with fungi. And when the beetles move on to a new place, they take their fungi with them. So they have little tiny pockets somewhere in their body, and those pockets contain the fungus that they coexist with, and they'll take that fungus with them to a new space and start a new culture of the fungus with it. As well, fungi fruit bodies can be direct food for a whole range of very specific beetles, some beetles that are found nowhere else except in polypore fruit bodies. They are also pathogens. This is a large, very common species in Northern Australia called Ganoderma statanum, and it occurs on a wide range of trees, often legumes, so acacias, poinciana, rain trees. In this case, it's killed a Eucalyptus cribra, which is an ironbark, and so they can be quite nasty pathogens for several of those species. So the main point of my talk today is a real problem that we have with polypore identification, which is that a lot of polypores all look the same. So in these 7 photographs here, there are 2 orders, 6 families and 6 genera, and yet they all look more or less the same. The fact that they have a fairly simple flat fruit body with pores on the underside allows for convergence, and that form can occur and evolve numerous times throughout the tree of fungi. So we have to try and use more subtle characteristics to try and identify these things. Microscopic examination is critical to try and identify fungi in the first instance, and you can see here the fruit body of the fungus. In this case, it's a Panus species. The internal flesh has both generative hyphae, which are these really thin things that you can hardly even see on the screen, and skeletal hyphae. And these are thick-walled. These ones are actually completely solid, so they don't have any contents inside the cell. They don't continue growing. They just grow out into the fruit body, and they're quite stiff and strong, and they give the fruit body its structure and its firmness. And depending on how much this vegetative skeletal hyphae that they produce, depends how solid and tough the fruit body will be and how long it will persist for. This finer generative hyphae does all the growing and eventually produces basidia and spores. Whether these generative hyphae have clamp connections or not is critical in the identification of polypores. You can see here, this cell is actually growing in this direction. It's formed a cross wall, and then it's passed some of its nuclear DNA back across this, what's called a clamp connection, back to the cell in the past. Those eventually make the basidial cells, and those basidial cells will eventually produce spores, which you can see down here. And there can sometimes be sterile cells embedded within that hymenium that are probably there to protect the spore layer from grazing arthropods like springtails. So in order to identify something, if you really don't know what it is, you need to get into that microscopy. Once you've identified something and worked out what it is, you can often identify larger macroscopic characteristics that you can see with the naked eye. And once you've worked out the diversity, then you can inform other people how to identify this thing without the need to go through all this complex microscopic analysis. Microscopy is quite tricky, and this is a major reason why our polypores haven't been sorted out in Australia yet. It can be very difficult to try and separate this flesh into its component cells, and often what you get is this, and you really can't see what's going on there at all. You need to start splaying the cells aside and start seeing some of the structure that's here. And here you can see a cross wall that doesn't have a clamp connection. And knowing that tells you a lot about what your fungus is, or more importantly, what it isn't. When you go a bit further and smash apart those cells even more, you can start to see individual spores and individual cells and the branching patterns that are produced. And looking around it enough, you can see different kinds of cells. Here we have some generative hyphae with clamp connections. Here we have a skeletal hypha that's unbranched and coloured. Here we have a binding hypha that is colourless but branched. And here we have an odd coloured spore in amongst the normal colourless spores. So all this kind of information is vital for getting an accurate identification and right down to species level. So what do we need to do in Australia to work out what polypores we actually have here and how to identify them? Well, ultimately, we need to get to field guides with correct names. A lot of the names that you'll see in field guides are unfortunately now quite outdated by sometimes 20 or 30 or 40 years. It is the case that not all polypores can be identified on site, but I reckon probably sort of half to 2/3 of them probably can be. And that's what I'm sort of working towards and trying to assist. Other species will always require that microscopic examination, but sometimes we can identify things by the host and the distribution of where they are once we eventually have all that information. I'm working on an interactive lucid key to Australian polypores, which is by far and away the easiest way to try and identify things, because you don't have to try and follow key characters. And if you don't have a character, all of a sudden you can't follow the key anymore. It allows you to choose whatever character you like and start from there. And I'm slowly building that. I was hoping we might have a draft version for at least people to try today, but it's still going to take me a while to code a few more before it's really useful for anybody. But before we do any of that sort of field guide level identification, we first need to go through the scientific characterization of all of the old species that we think we have in Australia and any new species that might be there. So we need to know how many species there are in Australia. What is the variation within those species? Which of the Australian names we're currently using are actually correctly applied? And that needs both DNA characterization and that microscopic characterization and comparison to the global literature, which is literally thousands of papers. And polypores can have very wide distribution, so we actually need to compare the entire global literature sometimes to identify a fungus. We need to identify any new Australian records of overseas names. There are quite a few things that have been recently described from Southeast Asia that may well occur in Australia. We need to identify if there are any undescribed Australian species that need to be described from scratch. There could be undescribed Australian genera, so the larger grouping genera are groups of species. We have a lot of genera that are only found in Australia in our plants, and we may well have quite a few genera that are only found in Australia in our fungi, particularly those that only grow with Australian plants like eucalypts. We need to know which species are introduced from overseas. We have a lot of plants that have come in as weeds, so we probably have a lot of weedy fungi as well. But at the moment, very few fungi are identified as introduced. We need to know which species are pathogens and what they're actually doing to our trees and our plants. And once we've identified what species we have, we need to know how to distinguish them all. And then finally, we can work out which ones are actually identifiable on site that the general public can easily recognize. So there's an enormous amount of work to do. And we've got that for some of our species, but nowhere near as many as we need. So I'm going to give you a little bit of a feel for just how much diversity there is and how much we know. This is what's called a phylogenetic tree that's made with DNA sequences. And these trees show the relationships between Australian polypores. This is a tree of almost 200 samples of Australian polypores in the family Polyporaceae. So they're the core polypores. And this tree shows just over 100 different individual species. They're grouped here by genera, and some things you might recognize the name of. So Ganoderma, Trametes, Perenniporia you might know, Phaeotrametes, but there's a whole lot of other things that you probably don't know, like Neophomatella, Pisipes, and these are things that have been more recently described. So this is sort of an overview. These are only Australian samples. So all of these things are definitely in Australia. We've confirmed that the genera occur there. These blue samples that are indicated by the blue arrows, they are new Australian records. So these are species that were not previously known to occur in Australia until I started examining them and looked at their DNA sequence. As you can see, there's a whole bunch of things out of that just over 100 species that are new Australian records. But at least they do have names. These brown arrows indicate the position of undescribed species. So far as I can check, I haven't chased every single publication in the world to identify these, but as far as I can tell, these are undescribed species. So they may occur in Asia as well, but at the moment they don't appear to have names. These red arrows indicate the position of genera, whole genera that don't appear to have names yet. So some of these, again, probably occur throughout Asia as well, but at the moment they are things that don't have even a genus name. So we have an enormous amount of work to do. It can take years to describe just one species, let alone the extreme number of things that we have to do here just in this one family. As a couple of examples, this is an unreported species from Australia. This is a thing called Chlamydon jupitivus, and it's known through Asia all the way to India. And it's a very beautiful thing. It looks like a flower developing. And these rosettes can be up to 1 meter across. They're absolutely huge. Sometimes they grow from the soil, from a clod of hyphae binding the soil together. Other times they grow on rotting wood, as you can see on this sign. They're not really true polypores, but they have little teeth on the underside. But their closest relatives are polypores, so they're derived from polypores. So I include them in the short term at least. I'm now, just in the last year, I've found 4 different locations from Babinda, Cairns, and up in the Daintree. And so this species is fairly common, but as far as I can tell, it's never been recorded in Australia, at least under this name. This is one of the apparent undescribed genera. It looks very much like a Moraderma, if you're familiar with that genus, but they're much, much smaller. They're only 1 centimetre or 2 across, and the pores don't bruise red like the Moradermas that we have elsewhere. And as far as I can tell, this is an undescribed species and an undescribed genus. It's quite common across Northern Australia, but you only ever find a couple of fruit bodies at a time. And as far as I can tell, it's just undescribed. Another group of polypores is the brown rot polypores. And you almost certainly will have seen this while out walking in the bush, that some wood rot has this brown cubical structure to it. And this is a specialized group of polypores that specialize in decaying the cellulose and hemicellulose, but they don't decay the much tougher lignin. So they're going for the easy bit of the wood to get rather than the hard bit. So they're sort of like fast food fungi. White rotters, in contrast, decay the cellulose and hemicellulose and the lignin and sort of decay to this white powdery stringy structure. So this very large tree over here is a phylogenetic tree of all of the brown rot fungi that have ever been sequenced from all over the world. This is looking at global distribution. Each one of these coloured boxes is a different genus, and the arrows here indicate the position of the Australian brown rot fungi. So you can see the Australian brown rot fungi are a complete subset from the diversity right the way across the world. But a lot of what we have is not what we think it is. The black arrows indicate species that are described species in their right genus. The blue stuff indicates undescribed species. The red arrows indicate undescribed genera, so whole genera that have never been described before. So we really have a lot of diversity in Australia that's just gone unnoticed up until now. The two brown arrows here indicate two introduced species of brown rot polypores, and we can be very sure that they're introduced because they only occur on pines, or European or North American pines. So one of the major findings of this particular study is that our fire fungi are not a single group as we thought they were. We call them all Lacocephalum fire fungi, but they actually come out in 5 different parts of the tree. So this is a fire fungus, if you haven't seen it before. They fruit within a week or two of a fire going through. So in this bare black earth, this giant thing will suddenly pop up out of the ashes and start spreading their spores around, ready to take advantage of this dead wood. They can do that because they have this large structure underground, which is called a sclerotium, and it acts like a tuber, and it stores all of its energy in there. And when they get the signal that a fire's gone through, they just pop up and start producing spores. They can sometimes, if you dig up these sclerotia and take them back to an office, they can sometimes just spontaneously fruit from a bare sclerotium, as this one has with Neil Boucher and Kevin Griffiths over in WA. So it turns out that the original species of Lacocephalum, Lacocephalum basilapaloides, which is only found in the Mallee country in South Australia and Western Victoria, it's called a stonemaker fungus because it makes these little fist-sized stones, and farmers, when they were first clearing the land, would come up with these fist-sized stones, and they couldn't understand why these stones were there, and it took many years for them to work out that it was actually a fungus that was doing it. It turns out that it is not even part of the brown rot group. It's outside of that. It's more closely related to Polyporus. The other 4 species of Lacocephalum come out, they are brown rot fungi, and they come out in different parts of the tree. So this Lacocephalum tumulosus is different to this Lacocephalum monomyleti. And these differ in their sclerotial form. So some of them are sand binding, they just bind a lot of sand together. Others have quite a large hyphae mass, which is more like a dough when it's fresh and then sort of becomes a bit harder. And this Lacocephalum monomyleti is related to what's called the native bred Lacocephalum myleti, which is quite a large fruit body, and it was eaten by Indigenous people in Southern Australia. And thank you very much to the people who provided images of these. So we have a lot to learn about our fire fungi, and what's particularly interesting here is that the Australian landscape has independently evolved this habit of fruiting into recently burnt landscapes independently multiple times. So this was a niche that was waiting to be filled, and there's a whole lot of things that have independently filled it. And our assumption that just because they had sclerotium that they would all be the same genus and have a single origin has been shown to be completely unfounded. Some other beautiful brown rot fungi. This is one of the fungi map targets. It's a thing called Gloriophyllum concentricum, except that it turns out not to be related to other Gloriophyllums at all, but part of this Polyporales brown rot clade. They form these beautiful shelving layers, and underneath they have gills, but instead of being radially lined like a mushroom, they are concentrically aligned. And it's one of only about 3 mushrooms worldwide that has, or 3 fungi worldwide that has these concentric gills. And this does occur at least rarely down in your neck of the woods around Brisbane, but it's more commonly found across Northern Australia. This beautiful fungus is very, very common across Northern Australia. And last year, Wayne found this down near Brisbane. And this is a eucalypt specialist. It specializes in heart-rotting eucalypts. So when you see eucalypts that are tunneled out inside, that's partly due to this species and a couple of others. These can be up to 40 centimeters across, and when they're fresh, they're this beautiful flame bright orange. It looks very much like a genus called Letoporus, and it's in the same family, but it turns out to be very distantly related to Letoporus, and its closest relative is actually a thing that's only recently just been described from Africa called Kusagiporia. So that's an Australian specialty. So as you can see, there's an awful lot of work to be done, even at the genus level, the deepest levels of our fungi. But once we've sorted out the genus levels, we'll need to start digging down into the individual species levels and work out how many species we have and how do we tell them apart. This is a study that I've been working on for the last couple of years with Teresa Labelle, now in Adelaide. And this is a study of the scarlet bracket fungus in the genus Pycnoporus or Pycnoporus. So it was thought that we had 2 or maybe 3 species in Australia. The names Pycnoporus coccineus, Pycnoporus sanguineus, and Pycnoporus cinnabarinus have all been used in Australia in the past. And it turns out to be both simpler and more complex than we thought it was going to be. The blue samples here in the tree represent Australian samples, and the black and red samples here are samples from elsewhere across the world. So this is a global tree of all of the diversity in this Pycnoporus group, which had never been examined in such detail before. This is a 7 gene phylogeny with nearly 9,000 line base pairs. So we can be absolutely confident of our results here, which is great. So I'm going to go through some of these species for you and give you an idea of their distributions. This is a beautiful little thing called Pycnoporus puniceus. It's found around the world, but it's quite rare everywhere. Until we started working on this, it really hadn't been confirmed to occur in Australia at all. And so we know it from now about 6 locations, because in the last couple of years, there's been a couple of collections made from up in North Queensland. It's a tropical thing, so it doesn't appear to get down into the southern Queensland, although it may possibly occur there very rarely. It has larger pores than the other species of Pycnoporus, and the fruit bodies are often quite small and often quite deep dark red in colour, and they often occur on quite small sticks. There is some genetic divergence between these different populations, and it's quite possible that there are actually different species amongst this Puniceus group. It's been suggested elsewhere in the world that these are not just one species, and we appear to have a similar range of diversity in Australia. This is the species that you're probably most familiar with. This is Pycnoporus coccineus. It occurs pretty much throughout Australia, but it's very rare in the tropical areas north of about 18 degrees. It's the most common thing that in fact, it's the only species as far as I know that occurs down in southeast Queensland. I thought there might be some Pycnoporus sanguineus down there, but everything that I've sequenced with small fruit bodies all turns out to be Pycnoporus coccineus. It also occurs in New Zealand, and it occurs all the way through the Pacific Islands as well. Interestingly,
I've collected sanguineus from southeast Queensland, Matt, yes.
Sorry, say again?
I've collected sanguineus from southeast Queensland.
Excellent. I would like to get some more information from you then.
Okay, very good.
Interestingly, this coccineus has also been recorded from China and Eastern Europe, but that turns out to be a sister lineage, but quite a deeply divergent sister lineage. And it's possible that these are actually 2 distinct species, and that's also been suggested before. But we have real coccineus, so it won't really affect our name. This is Pycnoporus sanguineus. The fruit bodies are much thinner than coccineus, and the upper side of the fruit bodies don't fade to the same extent as coccineus does. So young fruit bodies of coccineus can look quite bright, but as they age, they usually fade and eventually bleach white, and you often see sort of pale orange to white fruit bodies, and you turn them over and they're very bright orange underneath, and that's definitely coccineus. Coccineus doesn't really have any zonation on the cap. It's usually very smooth. I haven't shown you a picture here of sanguineus, but there's often at least faint zonation on the caps of sanguineus. The form of sanguineus that we have is the old world form, so it's the same as things through Southeast Asia and Africa. But things from the New World, from South America, actually are genetically a bit divergent, and people have suggested that these actually might be a distinct species as well. There's an African species here that had never been picked up before, that have been confused with sanguineus in the past, but it's clearly a distinct species. That's not something I'm going to describe, but someone else is going to have to go and look for that. This strange looking fruit body here we thought might be a new species, but it turns out that it's actually part of the true Pycnoporus cinnabarinus clade, which is otherwise only known from the Northern Hemisphere, through Asia and North America and Europe. And we only know this from one single fruit body near Darwin, and we're not really too sure what's going on here because the fruit body doesn't really form shells as cinnabarinus should. And also doesn't have clamp connections in the hyphae, which is the only individual fruit body in the entire, not just Pycnoporus group, but the entire Trametes group that doesn't have clamp connections. So it's a very strange thing. And we hypothesized that it might be something that has just blown in or blown in the past, and it's never found anything else to mate with. And all it's been doing is just producing these fruit bodies and maybe producing asexual spores rather than sexual spores, and actually being able to produce a normal type fruit body. Then we have this rather unexpected thing, which is only found in the Italy. It's a deeply divergent lineage, sister to this cinnabarinus group. And within it, there are actually two deeply divergent groups that may actually be distinct species. Every gene says that they're quite different from each other, which is very unexpected from such a small distribution. But at the moment, we're only going to describe a single new species here, and we're going to have to find more material before we can work out what's going on with the other lineage. So these look quite like Pycnoporus coccineus. They have quite thick fruit bodies. They don't have any zonation on the cap, but they don't fade to the same extent as coccineus normal do, and they have a real tropical distribution. So out of this study, we've confirmed the distribution and confusion around sanguineus and coccineus. We've found a new record for Australia, two new records actually, and then a new species or possibly two new species from the north. And so Australia really has a very high diversity of Pycnoporus, probably the highest in the world. So what does all this information tell us about the Australian polypores? Well, based on the information that I have so far, obviously we still have a very long way to go. I have to do at least a preliminary estimate of exactly how many polypores we have and get some idea of their diversity. So I've confirmed at least 550 polypores and polypore relatives for Australia. And I estimate that we actually have around 700 to 1,000 species. So we have a very high diversity of polypores on a world scale. Europe, for example, has around about 450, maybe 500, and a similar number in North America. And they are found in at least 160 genera. So if you're confused by the polypores that you're seeing, there's really good reason for that. It's very clear that many of the species in Australia are undescribed. There are many new records for Australia that need to be confirmed and identified, especially in the tropics. Many of the species, even the things that are new records, are just not illustrated anywhere. So you just can't track these things down on the net and identify them from images. And we have to go back to first principles. Our flora has a very strong division into temperate species and tropical species. There's not a huge amount of overlap. Unfortunately, where you sit in Southeast Queensland, you are part of that temperate tropical overlap. And so you will have species coming in from both directions. And you have a very, very large polypore, or potential polypore flora in your area. Most of the species, unfortunately, have unresolved classification. So unfortunately, quite a few of the names that we currently use will change. And that's just due to the need to update everything to be actually using current names. Even a lot of the names that we currently use need validating. Even though we understand the species very well, we need to understand that it really is that species and there isn't another name that we should be using, something from Japan or China that actually is exactly the same thing, and whether the genus name is the correct name. It's a little bit harder to work out how many of those species are only found in Australia, what's known as endemic species. But based on what I currently know, I estimate that it's somewhere between about 5 and 30% endemism. So 5% is at the lower end, but it's still quite a significant number of species. 30% endemism would be a very significant endemic ratio on a global scale. So that's the high end is probably not likely, but we certainly do have a lot of things that only appear to be Australian species, even tropics. Part of that is due to host specificity, things that only occur on certain species or certain genera. We don't know for many species what their hosts actually are, and we need a lot more observations to try and confirm this, even for things that we may have made 10 or 20 observations on a certain species. Our 21st record might turn out to be something on a different host again. So we need quite a few records to confirm host distribution. Some polypore species only occur in association with one species or genus. There are quite a few species that only occur on eucalyptus, for example, or eucalypts, including Corymbia, or on acacia, such as this Phalanus species. Most species grow on more than one host, what we call generalist, but most polypores probably are not completely generalist. They probably don't occur on every single bit of wood, but have an array of things with a lot of host jumps over their distribution. When we look at an individual block of wood, most of that wood that's rotting has several species of fungi that are coexisting in the wood, and only some of those are polypores. There's a whole range of different wood rotting fungi out there, not all polypores. But it may well be that the wood rotting community is unique to a particular host species. So while any individual species might occur on a range of different species, that combination of species may only be found on that one particular host. And of course, many polypore fungi are pathogens. That is, that they are detrimental to the tree that they grow on if they're growing on living trees. This is a thing called Ganoderma boninense, or basal palm root rot or palm butt rot. It's a devastating pathogen of oil palms throughout Indonesia, and it's costing billions of dollars for oil palm industry, which is tragic because they're cutting down rainforests to plant these oil palms that are then being devastated. We have reported this in Australia now on a range of different palm species. It's usually in gardens, but last year it was detected for the first time up at Cape Melville, a long way from any human habitation. So whether that's a natural dispersal or has been assisted by its spread through Australia, we don't really know, unfortunately. Polypores in forestry can devalue the timber, particularly with their heart rots. They completely take out the most valuable wood and render it useless, except for wood chips. They weaken limbs, so that's a very big risk for public parks with trees. And it turns out that many of our Australian species grow on living trees, but for a lot of them, we just have no idea whether they're actually pathogens or whether they don't have very much effect at all. As an example of some of those pathogens, this is a thing called Phalanus noxious. It's now been put in its own genus Pyrrhoderma, and it turns out that the species we have here is probably actually not noxious at all. So it actually needs a new name. It's a devastating disease. It occurs on at least 200 species of trees. And you can see they form this brown stocking at the base of the tree before they start fruiting. And you can see this is a hoop pine that's been completely killed, and there's quite a few trees in this plantation that have just been completely killed by the spread of this Pyrrhoderma noxion. This is a pathogen on a fig in a street tree in Cairns. It's a thing called Pomophagus colossus. And it's related to Ganoderma, if you know those. And it looks quite like it, just with a paler surface. And you can see cutting into the flesh that the pore layers here, which has the spores on it, are brown. And one of the defining characteristics of the Ganodermas is that they have brown spores. And these do have brown spores like Ganoderma, but their flesh is white or to cream and soft and doughy. It's like a dough ball. You can depress the surface quite effectively. And that's very different from Ganoderma, which are woody, hard and dark brown, at least at maturity. The pores don't bruise red very much. You can see they've been flattened here and they're really not bruising red. Ganoderma will sort of bruise red-brown if they're fresh immediately, and these don't. This is a thing that has been recorded once in the islands off the Northern Territory, but was not really confirmed for Australia, and was certainly not known to be a pathogen in Australia. This is another species that has only just been detected in Australia. It's known through Southeast Asia. It's a very voracious pathogen. It causes a heart rot that grows very actively. These fruit bodies can be up to 50 centimeters across and can grow within a matter of weeks, and they're very, very dense and solid. It again looks quite like a Ganoderma. It bruises reddy brown on the pore surface when touched or bruised like a Ganoderma, but the spores are white, not brown. And you can sort of see the white dusting of spores here. And it produces enormous numbers of spores. So when you're standing in front of the fruit body, you can just see like a mist of spores just blowing out from underneath it. The flesh is very, very, very tough and rubbery. It's like trying to cut reinforced chewing gum. You need a very, very sharp knife to get into it. And this mostly occurs on figs and a tree pea called Millettia pinnata or Pongamia. At the moment, we only know this from Cairns, but I've seen photos that may well be this from quite a wide range of towns across Queensland. So I'm very interested in more images of this, particularly if you can cut into the flesh and confirm it. This is an introduced polypore called Amyloporia sinuosa, which occurs only on pines, and it's quite a voracious decayer of pine bollards. And you can see this occurred on one of these bollards here, and it completely wrecked the bollard, and they had to replace it with a steel structure. It's a racupinate, but it can be identified by having a brown cubical rot, and occurring on pine timbers. So there's an enormous work to do before we really map out and understand the full diversity of our polypores in Australia. So how can you help, and how can you get identifications of your polypores? Well, as I said, Southeast Queensland is a key tropical temperate overlap zone, and you possibly could have many, many hundreds of species. And I would strongly recommend, if you're not already doing it, to be posting your pictures to iNaturalist. And in particular, the page that I curate, which is Polypore Fungi of Australia. And you can find that just by putting in Polypores Australia on iNaturalist. So the location data on iNaturalist contributes to the Atlas of Living Australia, which includes all of the data from herbaria and validated specimens. So when you post your specimens to this, you're adding dots on the map that are contributing to the Australia-wide Atlas, which is fantastic. But we definitely need photographs of all parts of the fruit bodies to be positive of the identification. It's not really good enough just to show a picture from above, because we really can't identify it from that. And it's also important to remember that you may not get an identification on something that you post, but that may just be that we're not able to do that right at this point in time. As our knowledge improves, we may be able to come back and identify that. So don't be disheartened by the fact that you're not able to get an identification right at this point in time. We're still learning. Ideally, we ultimately need specimens that are collected for a herbarium, so a permanent record, and ideally with DNA sequences. And I have some people around Australia who are able to send me specimens, and I'm able to sequence them and contribute to my research and the growing database on Australian polypores. And it's particularly important for you, where you are in Southeast Queensland, to be contributing to this kind of project, because there are many what we call type specimens, which are the original reference material for the names of fungi. They come from the Brisbane Toowoomba area, from the days of Frederick Bailey back in the late 1800s. And he collected quite a few polypores that he sent off to Kew, mostly in England. And they are the type material. But we've never sequenced anything from that same area. So we don't know when we apply those names elsewhere in Australia or elsewhere in the world that we're really getting the thing that he originally saw. So we need more material from Southeast Queensland that is associated with sequence data that we can then be mapping and tracking the application of that name. So I'm going to give you a brief overview of the things that you really need to be recording when you're taking photographs of fungi, particularly if you're putting them up on iNaturalist. So the first thing we need is a general overview of the fruit body, in particular showing how it's attached to the surface of the substrate and what it's attached to. The differences between fruit bodies, they can be shelving, what we call pileate, and broadly attached at the base. Sometimes they can then produce a bit of a foot down here, and this is what we call effused reflexed. So they are effused or smeared over the surface, but they also have a reflexed part, a little shelf at the top. If that shelf is lost, they become what we call resupinate, which just means lying on the surface. And those are the sort of three main forms, but sometimes they can actually have a little stem, and that stem can be central in the fruit body or lateral on the side of the fruit body. And so those five are the main different types of fruit bodies. So it's very important for us to know what they're attached to at the base, whether it's on wood or on soil, and exactly how it's attached. So a photograph from above and from the side and from below are all sort of necessary to capture that shape and attachment. There are also some other attachment features that are important to record. This is what's called rhizomorph or root forms on the side of the fruit body. It's most often found on the resupinate or crusting species, and they form these little root-likes across the surface. And there's only a relatively small number of things that have that, so you're well on your way to an identification if you are seeing these forms and recording them. Sometimes the shelves can be in aggregated structures and shelving in layers, what we call imbricate. And it's very important to record this if it's present, because again, there are only a relatively small number of groups that do this. So if you just pick off one of these fruit bodies and photograph it on its own, we lose that ability to know that it was growing in clusters. And that's something we could have used to get an identification. The cap surface is obviously very critical. We can sometimes have a cuticle, this red-brown layer that grows out from the base into this white margin. It is diagnostic for a thing called Earliella scabrosa, which is one of the most common tropical species that we have. In other species, that cuticle is black and grows out from the base and eventually forms a hard crust, like this genus called Neophomitella, which is actually very common, but had not really been reported from Australia until the last couple of years. In a very few species, they can have scales that are the remains of a layer that broke up from when it very first started to develop. And they are diagnostic for a few things like this Brasidolia uda, which used to be called Polyporus udis. Other times the surface can be very distinctive, such as this very shaggy cap, which is not found in very many species at all. Hairs on the cap are found in more species, but they're still very diagnostic for certain things when you see them. Quite a lot of species have what we call a screw post surface. These are much harder aggregations of cells, and they form like when you're pulling up a little tuft out of a meringue, but they set quite hard. And that can also be diagnostic for certain groups. Sometimes the cap can be splitting and cracking, what we call rhimose, and that's diagnostic for certain species, particularly in the family Hymenocutaceae with brown flesh. Sometimes the surface is varnished, or what we call lacate, and that's diagnostic for certain groups of Ganoderma, a very important one. And quite a lot of species are zonate, but it's again important to know whether they are or not. Okay, now the most important thing that I'm going to tell you here, the most important take-home message, is that we really, really, really need to know the colour of the flesh internally in the fruit body. It's something that a lot of people don't do when they're photographing, but often it's impossible to identify without knowing the colour of the flesh. A really good example is this one in the centre, this Coreopsis strumosa. The surface just looks pinkish to white, and you have no idea that the internal colour is this dark chocolate brown. So from externally, there's really no way you can identify this thing. As soon as you see this dark brown colour, you're immediately down to about 1 of 5 species. So it's very, very beneficial to take a photograph of the flesh. If you don't want to actually pick the whole fruit body, then just break a little bit of a chunk off the side. If it's still actively growing, it will just sort of continue growing over that and heal it. If it's not actively growing, then it's already dropped its spores and it doesn't really matter anyway. So what you can see here is a range of different flesh colors, from white to cream to pink to brown to orange to sort of pale creamy orange to dark brown to pink to dark vinaceous. And that's it gives us, it's one of the most reliable characters for identifying polypores. The flesh structure is also really important. This is what's called a duplex flesh layer. So the hyphae down here are radially aligned. They all head out towards the surface, towards the margin. The hyphae up here are all tangled up amongst each other. So it's a different structure and a different feel. This is all dense and hard. This is sort of very soft and depressible. And you can see here, there's a dark line of agglutinated cells that demarcate these two zones. And again, there's no way you can tell this without cutting into it and sectioning it. Very few species have what's called a granular core. They start to break down as they age internally and grow more hyphae around the broken down cells. And if you see this internally, you know you're down to one of only a handful of species straight away. And this is an example of an Inocutus species. This is an example of a surface crust on Ganoderma. You can see up here the black arrow, and that's sort of a very hard layer. And when you try and break it, that sort of cracks and crackles. And this particular species of Ganoderma, Ganoderma australe, has these what's called horny inclusions, these sort of little tougher nobules that form internally in the flesh. And that's diagnostic for that particular group of Ganoderma. Texture is also really important. You will have seen really hard woody species. Other species are fibrous. They can be cheesy, crumbly, or very fleshy, such as this beefsteak fungus. A few species can even have gelatinized flesh. This is an undescribed species in genus, as far as we've been able to work out, that has completely gelatinized flesh. It looks like a tube. This is a species of Gloeoporus. The flesh layer itself is actually quite soft and breaks up easily, but the pore layer is jelly-like, and you can pull it out for sometimes 5 or 10 centimeters. It's quite astonishing. The fertile surface is obviously also critical for you to photograph it. They don't all have the same shape pores. They can be at different sizes. They can be round. They can be elongated. They can even be maze-like or labyrinthine. They can be gill-like with branches, or completely gill-like, or even concentric and gill-like. And it's absolutely critical to get the hymenium surface so that you're able to judge that. There are poor relatives that have some strange features, such as this wrinkled form, sort of flattened teeth, plates, or just normal teeth. And these are sort of polypore relatives that are sometimes included in polypores and sometimes not. The pore-bearing layer stains a lot of the time. You will have seen what used to be called a Amoroderma. It is now called Sanguinoderma. True Amoroderma is only now found in the Americas. And what we have, the red staining ones, are now called Sanguinoderma, which means blood skin for obvious reasons. Ganodermas bruise red to brown. This beautiful little Physiosporinus initially stains lime coloured, and then it turns sort of rusty coloured, then it turns bright red, and then as it ages, it goes grey, and it ends up, when you dry it, having completely steel blue on the underside. It's an absolutely astonishing species. And you may have seen the bluing or greying species of Postia caesia group. Here's an example of a yellow staining Perenniporia species. There's also differences in the size of the pores relative to the thickness of the walls between them. So in this case, the walls are thin relative to the pore mouths. In this Coreolopsis, they are quite thick relative to the tiny little pore mouths. And one last thing that is can be very diagnostic is what's called hyphal pegs, little things that stick out from the side of the walls. And there's not a huge number of species that have these, but when you see them, it can turn you from something that you just can't identify at all to something that you know within 1 or 2 species what it is immediately. Both these species were not reported in Australia until last year. This beautiful little thing called Grammothele lineata has black hyphal pegs that stick up vertically out of the surface of the gills. It's a sort of inconspicuous little crusting thing, but if you can look at it with a hand lens, you can identify it immediately because there's nothing else remotely like it. Okay, one last thing. If you're looking at these woody Hymenocutaceae with brown flesh, it's very, very important that you know the spore color. So look underneath the fruit body and on anything that could have caught spores, particularly spiderweb, and see whether you can see the color of the spores, because the different genera have different spore colors. And so these things, just from a photograph, you wouldn't be able to tell apart what they are. Looking underneath, you know this has white spores, and in this particular case with a woody fruit body, it must be a Phalanus species. This one has yellow spores, and together with a couple of other features, you can tell that that's an Inocutus. So it's always, if you can, try and find some kind of spore deposit underneath the fruit body if you can't make a spore print yourself. Okay, I'm going to stop there. Thank you very much for listening to me, and I hope I haven't gone too much over time.
No, that's been wonderful, Matt. Thank you very much for sharing that wealth of information. So I guess a few questions that we have. So the take home for me was really about flesh color. And I've had this really basic, and I think I mentioned this to you earlier on, this very basic understanding of flesh color. So what I would do is I would look at, and I'm just showing a picture of the Fomitopsis that I showed you. So again, this was a Fomitopsis that I was looking at, that I collected. And I noticed that it had a pink flesh, and I thought pink flesh is Fomitopsis. But I think you had something to add there, didn't you?
Yes, the genus Fomitopsis is quite complex. Real true Fomitopsis has white cream flesh.
Right.
Some people remove the pink fleshed things out of Fomitopsis into a genus called Rhodofomitopsis. Rhodo meaning rosy, for obvious reasons. So they're not so the true core Fomitopsis, and some species of Fomitopsis can certainly have white flesh. But when you see pink flesh like that, you're definitely down to only a couple of groups of polypores that have pink flesh. And that pink staining is also very useful to tell you that you have Rhodofomitopsis lilacino-gilbus.
Very good. The other thing I've been collecting in the field is I try and get this sort of shot pores per millimeter. Is that valuable information or?
Very valuable. That's correct. Yes.
So again, I can, if you're out there in the field and I can give you these tags, you'll see that I can gather from this that this thing has 2 to 3 pores per millimeter. So pores per millimeter here and flesh color can be obtained.
Yes, that's one of the most useful things for identifying as well, because polypores vary quite a lot in their pore size. And when you don't have much else to go on, that's absolutely critical.
And the other thing for me, my 101 basic information was that Trametes, and here is a picture of Trametes polyzona. I think it was actually you that gave us the ID from the field images. And I gathered that I took an image of the flesh color. So here it is. Here's a picture of me getting, and I thought that, so my basic understanding was pink was Fomitopsis. Now I know pink is Rhodofomitopsis, and Fomitopsis is white flesh. And tan flesh I thought was Trametes, but I believe that it's only the species of Trametes that has tan flesh. Is that right?
That's correct. Yes, there's only a couple of species of Trametes that have tan flesh. The others are usually white. Occasionally they can have a little black line.
Do you often have
but in this case, it's absolutely critical to show the flesh color here, because there are a couple of other species that look identical to this, and without the flesh color, it's just unidentifiable.
Very good. Do you often struggle with? There's a picture of that polypore that we found down here as well too. So again, there's a picture of the context and of the flesh.
Beautiful. Yes, that's a magnificent thing.
Do you have much success with getting spore prints from polypores, or do you struggle with that?
It's very variable. Polypores often have long-lasting fruit bodies that at least persist through the season, and they can be very choosy about when they drop their pores. That big orange one that you just showed, it took me probably 10 years to get a spore print from that, and eventually I had to camp on top of it and put a tape a glass slide underneath it with sticky tape, and it still took me about 5 nights to find a night that was actually sporulating to get a spore print on it.
Very
so
good.
It can be very, very fussy. There are a few species that you can actually pick and put in a humid environment, and they'll actually sporulate, even if they weren't in the field. It's usually some of the larger species that do that.
Is
So
it-
yes, it can be very, very difficult to find spores on polypores, which is one of the things that makes them very hard to identify. But when they're fresh and actively growing, if you can get them onto a surface, ideally a glass surface, because often they are white spored, but otherwise a bit of paper, that's your best bet.
Okay. So these are the two types of Pictoporus that I thought we saw down here in southeast Queensland. One of them is this Pictoporus cochineus, which is this sort of thing, and rather common. I believe also associated with Indigenous uses as a teether for babies.
Yes, at least in Central Australia.
Interesting. And this was the zoning, as opposed to the cochineus, which has that sort of, and again, I think people will know this from the field, that it's rather common down here underneath a brighter red than it is on top. And that's the top surface. And again, it fades a lot. But the sanguineus has that zoning. So this was what I understood sanguineus to be. And it has that zoning, that's it, that zoning on top, that zonation of colour. So that's a specimen we collected.
That certainly looks like sanguineus, yes. And you can see the fruit bodies are very thinly attached. They almost have that a little stem at the base because they're not broadly attached to the surface.
Yes, yes. All right.
That's fantastic.
I'll stop sharing my screen. How do I stop share? And I'll turn my video on. So some questions we had were, and I guess they might be sort of 101 questions, but we're talking about polypores. So I guess we expected to see pores, but you mentioned gills at some stage, concentric gills. So one of the questions were, what are gills? So I guess we have a lot of entry-level people who may be just cutting their teeth. So why were you talking about gills when we were talking about polypores?
Gills are any flat plate surface. So you can see a great example of gills in the picture behind you. They're surface that we know from mushrooms. But any flat plate is called a gill in mycological terminology. So that doesn't necessarily have to be radially aligned. They don't have to be only flat. Sometimes they can be forked, or they can be quite irregular or concentric in the case of some of these. So we're really, if it's a flat surface, it's a gill. If it's a round hole, it's a pore, or an odd-shaped off-round hole, it's a pore. If it's a projecting peg, then it's a tooth.
Very good. So the other question I had was about what is a good book for microscopy, and the one that I have here is called, and it's really more about mushrooms than polypores, but this is the book I have. It's called How to Identify Mushrooms to Genus 3, and it's on microscopic features. So if anyone's interested in that book, that is a good one for microscopy, generally speaking, I believe. But
I would very strongly recommend that book. That's the one that I had when I started to learn.
Okay, so it's by David Largent and David Johnson. So if anyone wants to email us, we can probably get hold of the book, or you might be able to get it online, I imagine. So that's a good one for microscopy. And the other thing was with regards to polypore flesh color and spore color, we often use a color chart such as this. So we'll use, rather than the subjectivity of saying something's brown or tan or pink, we sometimes recommend, so if anyone wants a color chart, they can email us at info, and they're $7.50 plus postage, and we can post you a copy. We have a limited number of these available, but we do get more. So if anyone wants a copy of the color chart, let me know. Probably first dibs will be given to members of the society. So happy to do that. I guess it can remove subjectivity from things such as color.
Absolutely. Do you want me to answer that question on Pycnoporus versus Trametes?
Oh, yes. Was there a...
It's one of the last questions there.
So I'll read out the question. What was the question? Could you read out the question?
From Peter. So Pycnoporus are not called Trametes. I should have probably explained this a little bit. This is an area of contention right at the moment between polypore experts, it's realized that Pycnoporus are nested within Trametes, and there are two possible solutions. Either we call everything Trametes, which is what some people are advocating, or that we keep Pycnoporus and Trametes get split up into several other genera, like Sclerodepsis and Artolenzites. And so it needs to be, there needs to be a bit of a consensus around that before we really have a solid way forward. And at the moment, people are arguing strongly both directions. So iNaturalist at the moment calls everything Trametes. And so all the Pycnoporus are Trametes, but other people are strongly resisting that. So that's a toss-up, and you can use either names in the years that it takes to come to a consensus.
So Pycnoporus is a valid synonym, so you can call it Pycnoporus,
yep.
And the name would be valid, or you could take the other camp and call it the Trametes variant.
That's right. iNaturalist, of course, has to go one way or the other, and they've gone with calling it Trametes, and so that's what they use. So,
okay, very good. All right. Does anyone have any other questions? Other questions. So you mentioned Basidio versus Asco. So I guess there's that 101 sort of introductory knowledge of the difference between Basidio and Ascomycete fungi. And I guess it's really, I always describe it as the division within, the initial division within the fungi kingdom. So, and it really has to do with microscopic spore dispersal structures, doesn't it? So Basidiomycetes have a Basidio structure as opposed to Ascomycetes, which have ascos or sock-like structures which exude spores. Did you have a brief overview of those sort of terms and the differentiation between them? Do you have many Ascomycetes and polypores?
There's nothing that's really a true polypore in the Ascomycetes. No, so what you said was absolutely correct. The Ascomycetes, it's a microscopic term, so you need to get down to the spore-bearing cells themselves. Ascomycetes have a sac, and the spores are held within the sac, and they are usually forcibly ejected out the end of the sac. So the cup fungi, for example, can forcibly eject their spores upwards, and so they can have their spore-bearing layer on the upper surface. Basidiomycete spore-bearing cells are a cell that produces little branches at the end, and there's a spore that sits on the end of those branches, and then those spores are released a fraction of a millimeter, and then they drop down and into the wind column. So Basidiomycetes have to drop their spores downwards, which is why the pores are on the underside, or the gills are on the underside, because those spores are not really shot forcibly, they're just dropped into the wind column.
So most polypores are Basidiomycetes.
Yes, everything that's a real polypore is definitely a Basidiomycete.
Very good. The other questions were, there was a question about why is fungi in its own kingdom. So I could give you a 101 sort of explanation. Is it fungi are neither plants nor animals. They're very different to plants, very different to animals, but they're probably closer to animals than plants because of their cell structure. And it has to do with the fact that the cell structure is generally made up of chitin, and chitin is what you find on the exoskeleton of insects. So that fact places fungi closer to animals than plants, but they are in fact neither animals and plants. They are in their own kingdom. So the third kingdom, but there are 6 kingdoms now. Did you have anything to add in that space?
Depends who you talk to. There are, some people say there are a lot more kingdoms than that.
Certainly, I'm sure there are. Other questions? We have a very specific question here from James Strong. Thank you. Do you recommend any specific primer sets or Sanger sequencing to complement ITS14 for I-Ding Ganoderma species?
Yes. Probably one that you should probably email me about, and I'll track down the exact details of the recommended primers.
Great, James. So anyone else too, if you have a very specific question that we haven't answered here, info@queenslandfungi.org.au. I've put that in the chat, and it's also in the back of my image up here. So if everyone can see that, if you email any questions there, I can send them on to Matt, and we can get you a better answer to any questions you have. someone's asked about, will they kill healthy trees or that have been compromised? So I guess they're talking about the presence of parasitic polypores on trees. And we often get that question about, you know, is this a bad thing when you see polypores or brackets or parasitic fungi on trees?
For most species, we don't really understand exactly how and when they get into the tree. They may have been growing for years or decades before they start fruiting, of course. And most of the time, we just see the fruit bodies as they're starting to finally decay, but we don't really understand the causal link between that initial decay. And of course, it very much depends on the species. For things like Phylinus noxious, they are definitely able to attack healthy, strong trees and actually decay them, and they slowly lose health over a period of years. For some of those Ganodermas, they definitely attack trees and actually kill them. And once you start seeing, for some of the things like Albitzias, the rain trees, when you see the fruit body starting to form, you know that tree only has a year or two left before the tree is dead. So they are definitely associated, and some of them are very definitely associated with attacking healthy living trees and killing them. A lot of the time, though, it's just a gradual decay, and it may be that that part of the tree has already been dead for a while, and the fungus is just growing on the dead wood. So just because you're seeing a fruit body doesn't mean that the tree is on its way out. It very much depends on the species, both of the tree and of the fungus. And of course, tree hollows are absolutely critical for a whole range of different animals. And if we just took down every tree that has a fungus on it that is creating that habitat, we'd actually lose a lot of habitat for animals. So it's very important to identify the species. And I'm working very closely, particularly with the Cairns Council up here, trying to work out this link between what the fungus is, what the tree it's growing on, and what that actually means to the tree over years, so they understand which fungi are actually not detrimental and which ones are actually going to are really a danger and they need to be actually taken care of. It's definitely not a one-size-fits-all answer.
Wonderful. Well, thanks, Matt. That's been terrific information. So get on board, everyone. Get on iNaturalist. Get into Citizen Scientist. Start specimen collecting, but don't forget that valuable field information: flesh colour, spore colour, how they attach, the top, the bottom, the in situ photographs, and of course spores per millimetre. All of that information. Thank you very much, Matt.
And thank you, Wayne. Thank you, everyone.
Thank you. All right, I'll stop recording now. Thank you.